Hello, everyone. Welcome to today's live broadcast, highly multiplexed single-cell analysis of tumor heterogeneity through time and space by mass cytometry. I am Judy O'Rourke of LabRoots.com, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Cell Signaling Technology. Cell Signaling Technology is a private family-owned company founded by scientists and dedicated to providing high-quality research tools to the biomedical research community. The company's employees operate worldwide from its U.S. headquarters in Massachusetts and offices in the Netherlands, China, and Japan. For more information, visit www.cellsignal.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can, and you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button located in the lower left of the window. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's first speaker, Randy Wetzel, Director of Cytometry at Cell Signaling Technology. Dr. Wetzel received his PhD in neuroscience from Boston University. He then worked as a research fellow at Harvard Medical School, Massachusetts General Hospital before joining Cell Signaling Technology in 2002. He is currently the Director of Cytometry at Cell Signaling Technology, where he oversees antibody testing and validation in imaging and flow cytometric applications. I will now turn it over to Dr. Wetzel for his presentation.
Thank you, Judy, and thank you all for attending this webinar today. I hope you find the content as exciting as I do. So we're talking about IHC today, and there are a number of different methods for this, whether you do chromogenic IHC, as on the left in this image, multiplex immunofluorescent IHC, as in the center image, or the very high-plexed imaging mass cytometry in this image from Bern on the right. Regardless of which of those you do, I think it's critical to remember that you have to use antibodies that are specific. And by that I mean that they bind to specifically only to the target of interest, and they don't bind to other proteins that might be present in the tissues. So the protein that's imaged in the brown on the left and in the green in the center is a well-studied protein called PD-L1. And if you were looking to set up a study like this, you might be looking out to find a PD-L1 antibody. And here is some chromogenic IHC data with two commercially available PD-L1 monoclonal antibodies. And as you can see, they both stain PD-L1 positive lung tumor tissue. However, the one on the right also stains PD-L1 negative breast tumor. So this is not PD-L1. So based on this information, you would hopefully choose the antibody on the left and not the antibody on the right. However, it's not always clear when antibodies are nonspecific. And often these antibodies are used in studies and then published, and subsequent research is based on those publications. And this has led to a big problem with irreproducibility. This is really highlighted by two eye-opening papers from 2011 and 2012, in which scientists at two different pharmaceutical companies, Bayer on the left and Amgen on the right, were looking at how often they had trouble reproducing published studies when they're working on their translational assays. And surprisingly, one found that 2/3 of the projects had inconsistencies between the published data and their own in-house data. At Amgen, it was even more startling. They were only able to confirm the scientific findings in the publications 11% of the time. So this means that there's a lot of irreproducible studies out there, and the cause of that really varies from, maybe not using proper positive and negative controls, maybe a little bias in the pressure to publish, but at least some of this is due to bad antibodies. It's been estimated that of the spend on protein binding reagents, approximately half of that is wasted on bad antibodies. This is about $350 million in the U.S., or the equivalent of $800 million US globally. So this is a big problem. And it's a little confusing in the market space out there, because you see lots of companies talking about validation, but I think the biggest problem is that there isn't a clear definition. As this quote reads this is from a David Rim paper a few years ago, there are no universally accepted guidelines or standard methods for determining the validity of these reagents. So as you look around as a scientist, you see different vendor websites, and they have words about validation and testing. They may even have a stamp or a seal of approval. But as a scientist, I would urge you to be cautious about that and to do a little investigation, to look at who's doing the validation, what are the testers' qualifications, what were the testing models used, and most importantly, did they use appropriate positive and negative controls. So as Judy mentioned, my role here at Cell Signaling Technology is in testing and validating antibodies. And we have a pretty rigorous validation paradigm that I believe is pretty tight. So the key to it is concurrent testing on a number of different applications. So often an antibody will look okay on one application, and you won't see a problem until you put it on a different application. We also use target-specific modulation. So in other words, using different ligands or inhibitors to alter the activity, expression, or localization of a protein. And then we'll look at that and say, is this what I expect? We stain positive and negative cells or tissues when they're available, and when they're not, we can generate knockout or CRISPR models to really strengthen our validation. And equally important, as new lots of antibodies are released, they're retested in all of the applications again to verify that nothing has changed. Here's an example of some data that came in. These are real data that came, these 4 clones came up to our group for testing, clone A, B, C, and D. And they're shown here with Western data on 4 different cell lines each. And you can see they look pretty comparable. The molecular weights are about the same size. However, when we put these clones on just simple immunocytochemistry, you can already see differences in the staining pattern between the clones that's not apparent at the Western level. And so it's this combination of concurrent testing on multiple applications that really strengthens our feeling that the antibodies are specific. And I'm very proud of what we do, and I think it's recognized in the industry. In the most recent BioCompare Research Antibody User Survey, CST was ranked #1 in terms of antibody specificity, sensitivity, and reproducibility. And I believe it's from our rigorous validation paradigm here. Recently, this subject of antibody reproducibility was raised at a FBLI conference at Asilomar in California. This was a meeting toward the end of last year where experts from across the field, including antibody vendors and scientists that use these antibodies, met to talk about antibody validation and what's causing the problem. And as a result of that, a terrific paper came out right at the end of last year, published in Nature Methods, where the authors, it was a team of experts, described a testing paradigm that we should all use to look at our antibodies. And they refer to this as the 5 pillars of antibody validation. And at least the 4 on the left in this slide are relevant for what we're discussing today, IHC. So the first is genetic, and this can include things like using siRNA to knock out the target, and do you see your stain go away? Orthogonal studies like mass spec to confirm that the expression you see with your antibody matches the protein expression by mass spec. Also using an independent antibody, so using another antibody against the same target, do you see the same expression pattern and localization? And finally, if it's available, a tagged express protein to show that my antibody is capable of staining this protein. So it's the same kind of idea of using multiple applications at the same time to really solidify the validation of these antibodies. So just because an antibody is specific and validated, though, does not mean it will always work. Sometimes it's possible to get bad data with good antibodies if they're misused. And one example is not following the recommended protocol. So antibody performance can really vary if you use a different fixative or different conditions of staining, and it's important to follow the recommended protocol. Also, using the antibody in an application that is not recommended or supported. For example, if an antibody is validated for flow cytometry or immunocytochemistry, does not mean it's going to work in formalin-fixed paraffin-embedded tissue. Poor sample quality or sample preparation, as always, can negatively impact your results, or the quality of the other reagents that you're using in your stain. So with that, I'm going to pause and hand over the mic to my colleague in Zurich, who will show us his amazing data using imaging mass cytometry.
Thank you, Dr. Wetzel, for that informative presentation. And now I would like to introduce Dr. Bernd Bodenmiller, Professor for Quantitative Biology at the University of Zurich. Professor Bodenmiller studied biochemistry at the University of Bayreuth and ETH Zurich and obtained his PhD for his work on system-wide signaling network analysis in the laboratory of Rudi Ebersold at ETH Zurich. For his postdoctoral training, he joined the laboratory of Gary P. Nolan at Stanford University. There, he developed methods for the high-throughput analysis of signaling network states by mass cytometry, a technology that allows, in principle, to quantify 135 proteins and signaling molecules at the single-cell level. In 2012, he became group leader, and in 2013, SNF ERC assistant professor at the Institute of Molecular Life Sciences at the University of Zurich. Currently, his group is developing methods for highly multiplexed imaging by mass cytometry to unravel how transcellular signaling network interactions in the tumor microenvironment drive cancer development and ultimately might be exploited for therapeutic targeting. I will now turn it over to Professor Bodenmiller for his presentation.
Thank you for your nice introduction, and also I would like to thank CST for offering me or for providing me the opportunity to present some of the work that we are doing in my group here at the University of Zurich. So, as the title already says, this presentation will focus on the highly multiplexed analysis of tissues by imaging mass cytometry. So, it is very obvious to all of us that tissues are at the center of life science research, because any type of disease happens in a tissue. Either it originates in a tissue, or it nevertheless happens in a tissue, such like an infection. Now, one disease we have a particular interest in that is originating in a tissue is cancer. And you can see here on the slide that typically in cancer one can find different tumor cell phenotypes, as well as multiple immune cells and stromal cells that have infiltrated the tumor. And I think it is clear to everyone that in order to study such a complex tissue such as the tumor, but also any other tissue, what we would like to have is a comprehensive measurement that allows us to distinguish all of these cell types, as well as to infer the functional state of all of these cells. But now let's look at how these measurements are actually done, for example, in the clinics and in many research groups. So it is still routine work to do an H&E stain where one can see the distribution of proteins and nucleic acids to learn about the morphology of a tissue. And I'm always impressed about how much pathologists can tell me about a tissue simply by looking at such an H&E stain. Now, another technique that is extremely widely used is immunohistochemistry, where you see the distribution and the abundance of a single epitope, typically a protein. And I think every medium-sized hospital makes millions of those stains every year to assess the diseases of their patients. And then there's also, in some specialized labs and in very few hospitals, there's actually multiplexed fluorescence microscopy used, where 2, perhaps sometimes 5 markers are visualized at the same time. However, if you now think again about the tumor that I've just shown you, to truly comprehend its composition and to study it, what we really need are highly multiplexed measurements with spatial resolution of the tissue. And in an ideal world, what I would like to measure is the genotype, the transcriptome, the proteins, the signaling network state, lipids, metabolites, etc. And of course, in the moment, that is not feasible yet. But let's assume we can do highly multiplexed images of such tissues. Then we can also identify the boundaries of individual cells. Then we can learn about cell types and their states. And as we have an image, we can also learn about the local structure and arrangements and cell-to-cell interactions that happen in these tissues. Now let's assume we can generate these 3 levels of information. Then together with patient data, what this will yield us immediately is information about the underlying biology. Because compared to a proteomics or genomics measurement that is performed on bulk cell samples, here we not only get a long list of markers that are up and down regulated, but we immediately see them in the context of cell types, many of which we know, and in the context of other cells. So we can readily place what we see in the context of known biology. And understanding biology is always the basis for drug discovery, to improve existing drug regimens. And also, if we have such imaging techniques, we can also use it to assess the efficacy of a drug. And if we now assume that we can generate such images of a high number of patients, then I'm very, very confident that the information about many markers, cell types, and their states, and cell-to-cell interactions will be much more informative to stratify those patients compared to a single stain as it's done in IHC alone. And what I believe what will happen in the long term with this highly multiplexed tissue analysis method, and the one that I present you today is just one of several that are actually currently are being developed, is that these techniques will be used for a more accurate classification of the patients, to give them an accurate prognosis, to choose the best possible treatment, and along the way then also to assess the treatment efficacy. Now, in order to perform such highly multiplexed measurements, we need a technology that allows for highly multiplexed readouts. And in an ideal world, we also want to have those measurements in a targeted manner, meaning that we can predefine a list of proteins, phosphorylation sites, and other markers that we're interested in, and then we can measure them in a targeted manner in a tissue. So I think most of you have already heard about mass cytometry. It's in its original iteration a technology that is similar to flow cytometry, in that you take cells in suspension and then you stain those cells using antibodies. However, now what is unique to mass cytometry, and that is very important for you to understand the approach, is that an antibody with a defined specificity is labeled with a metal isotope of a defined mass. And this mass in the end will be the reporter that we read out. So then the next step, we inject those single cells into the mass cytometer. Here they enter so-called plasma. What is a plasma? It's the fourth state of matter. You probably remember from your studies, there's solid, liquid, gas, and plasma. You always have to increase the amount of energy you add to the system to reach a plasma. And I think the most famous plasma that we all know is actually the sun. And like the sun, also the plasma in the mass cytometer is actually pretty hot. It's about 8,000 degrees Celsius. And so when a single cell now enters the plasma, it is atomized, and most of the atoms are ionized. And this generates a single cell ion cloud, which is analyzed using a mass spectrometer. And the result is shown to the lower left in this slide, is that of every single cell, we measure the distribution and the abundance of the metal isotopes that were bound to the single cells via the antibodies. And we, of course, know for every isotope to which antibody it was bound to. And therefore, we can learn about the cell type, signaling, and functional state of those single cells in a targeted manner. And at least theoretically, the mass cytometer provides 135 simultaneous measurement channels. So why do we use isotopes as reporters? The most common reporters in biology are probably fluorophores. However, as you can see in the lower left of this slide, they have rather broad emission spectra. Once you measure several antibodies with fluorophores attached to them, those spectra start to overlap, and eventually it becomes quite challenging to tell, thanks to this overlap, apart at a given wavelength from which fluorophore the signal was actually coming from. Now, by replacing those fluorophores with metals and atomic mass spectrometry, we by and large solve the problem of the overlapping channels and now can measure many more molecules simultaneously on the single cell level. Now, in the second part of the talk, when we talk about imaging mass cytometry, you will also see that using metals instead of fluorophores or fluorescence as a readout also solves us the problem of the background fluorescence that we can find in tissues, and that is quite dramatic in some tissue types. So here the current status of suspension-based mass cytometry. In the experiments that we perform in my lab, we always determine the cell viability, the cell size and volume, the DNA content, and the cell cycle state. In addition, we can measure 52 antibodies simultaneously, and we can measure about 1,000 cells per second. And this measurement speed can be maintained over hours, so there's no problem to analyze millions of cells. The detection limit is about 100 copies of an epitope per cell, at least on paper. Then we can analyze hundreds of samples a day. We have developed protocols to do this, and we, for example, run screening experiments where we really analyze thousands of samples within a few days. And then there are a minimum of, I would say, 400 antibodies that now have been validated for mass cytometry. Now, the caveats or limitations of mass cytometry are threefold. First, the cells are atomized, meaning it will always and only be an endpoint measurement. So you cannot use it to sort cells and do follow-up experiments. We also do not gain any live cell information. And to understand many biological processes, this is actually important. And then we have to rely on antibodies. And as Dr. Wetzel already pointed out, the number of antibodies that is available, that is of high quality, reproducible, and highly specific, is in some sense limited. Or from my view, we don't have such an antibody for every single protein yet of the human proteome. Okay, so now what I've explained to you so far showed how we can use mass cytometry to analyze cells in suspension. And I would now like to come back to the question of analyzing, or the problem of analyzing a tumor or a tissue. We can do this with suspension mass cytometry, but then we have to generate single cell suspensions from a given tumor or tissue to then analyze it by mass cytometry. And when I started my group here in Zurich, I was thinking about how could you use mass cytometry akin to a microscope. And the idea that I got when I was visiting by chance a group here at ETH Zurich called Detlef Günther. When I visited Detlef Günther, he's a specialist for laser ablation technologies, that really occurred to me that the most feasible way to introduce a tissue akin to single cells into the mass cytometer would be using laser ablation technologies. And so what we then developed is what we call imaging mass cytometry. And in imaging mass cytometry, we take routine tissue sections, we stain those sections with the antibodies that carry the pure metal isotopes. Then we use a laser ablation system, and this laser then shoots on a very small area on the tissue, let's say a square micrometer. And in this very small area, all of the tissue is removed. We generate a cloud of very small tissue particles. And now this cloud of these particles is introduced into the mass cytometer akin to an individual cell. And then the process is, like I've explained to you before, however, now we determine the distribution and the abundance of metal isotopes, not for an individual cell, but for a distinct location on the tissue. And here I would like to stress is that the laser doesn't just shoot once and then we measure, but this laser continuously shoots with several hundred hertz and scans over the tissue line by line by line to generate the images images of the tissues that we are interested in. And all in all, we can now also image 52 markers simultaneously, currently in the tissue by imaging mass cytometry. So besides that, my lab has developed a complete workflow for imaging mass cytometry. This concerns the upstream tissue processing. Here we did some optimization. We also established standards to make measurements comparable among machines, among many weeks. We invested a lot of time in antibody validation, and I will come to that on the next slide. I've shown you how we then do the measurement. But then we also developed a broad computational toolbox to generate the images, to segment the images into individual cells, and then to do downstream data analysis. So now here, I quickly want to show you the types of antibodies that we have already validated. Of course, we are centered on the topic of cancer, and therefore we have antibodies that tell us if cells are apoptotic. We can distinguish many cell phenotypes, immune cells, stromal cells, also different tumor cell types. We know if cells are proliferative. We can measure many transcription factors that are informative about the cell's motility or the inflammatory state. Thus, we can also tell about the immune response. We can tell you the cell cycle state, or if there's a DNA damage and repair mechanisms are ongoing. Where we still have some troubles is in measuring the metabolic state of within the tissue by imaging mass cytometry. And besides that, we also have, I would say, about 40 antibodies that we have validated against phosphorylation sites, so we can nicely read out the signaling network state of individual cells within the tissue. And all in all, this sums up to about 150 validated antibodies that we now work with imaging mass cytometry. And at the very end, I will also show you or discuss with you on a couple of slides how we actually do the validation. So here now a quick history of imaging mass cytometry. When we started to develop this technique, it took us about 14 hours to image a square millimeter. And the image that you see here is from a breast cancer tissue. In green, you can see HER2, which is typically at the membrane, vimentin, which is normally in the stroma in pink, and H3, which we use sometimes as a nuclear marker, shown in blue. And you can see that this image is still kind of blurred. Then we improved the reagents, we improved the protocols, image quality became better, our approach became faster. Then we built a dedicated prototype imaging system, and at that point, it took us about 5 hours to analyze a square millimeter. But more importantly, the images started to look similar to what you will get with routine immunofluorescent microscopy. Please note on this image is again a breast cancer tissue section, how we can nicely demarcate individual cells and how HER2 is nicely bound to the membrane and there's no blurriness, etc. So here I'm often asked, where's actually the vimentin? And all this information is actually contained in the data that we generate. But in imaging mass cytometry, we have a very high dynamic range. It's about 10 to the power of 5, which means that you use the automatic min-max settings to visualize a marker. You're sometimes left with the impression that you couldn't detect a given marker, but then if you adjust the scale, you actually see that there's rich information contained in the image. And that is compared to IHC, or fluorescence microscopy, where you have perhaps a dynamic range of 100 to a few hundred. So since then, we further improved the technique, and by now, routinely, we can analyze a square millimeter in about an hour. And on this slide, you see a direct comparison between immunofluorescence microscopy, shown to the left, with imaging mass cytometry to the right. And to generate those images, we again took a breast cancer tissue section and we stained it with an antibody against HER2 and an antibody against cytokeratin. But before staining, we split the antibody, labeled half with a fluorochrome and the other half with a metal isotope. We mixed the antibodies again, stained the tissue, scanned it in the fluorescence microscope, which generated the image to the left. We took out the very same tissue section and put it in our imaging mass cytometer and scanned the same region. And this generated the image to the right, where you see in false color the distribution of the measured isotopes. So there are several things that we can now learn from this comparison. First, really the distribution and abundance of the measured markers is basically identical between fluorescence microscopy and imaging mass cytometry. And we also did this comparison for IHC, and we basically get the same result. You can also see that although our resolution in our routine settings is about 3-fold lower because we measure with a spot size of about 1,000 nanometers, while fluorescence microscopy, you're probably around 300 nanometers, we nevertheless can nicely see the membrane structure and also nuclei and the cytoplasm. However, in our measurements, we have information of over 40 markers simultaneously. Now, we also did quantification of the comparison between imaging mass cytometry, which is here on the y-axis, and immunofluorescence microscopy, which is on the x-axis. We did this for many markers, and we can really see that the R-square, so the correlation, is at least 0.8. Often it's higher. But the difference between the two techniques can be explained. So if you look to the lower right, you can see that there are many cells that are very high in fluorescence microscopy, but low in signal in imaging mass cytometry. And when we went back to those images to find out what the cause is, then the main reason was the autofluorescence of the tissue. And as I stated initially, in imaging mass cytometry, we do not suffer from autofluorescence. So now with our approach and the given throughput, we can also image larger areas. Here's the breast cancer tissue, where we analyzed 44 antibodies simultaneously, and you look at an area of 4 × 2 millimeters. And I just quickly, in a minute or so, show you how heterogeneous and complex actually a tumor can be. And so what you see in a moment is an unspecific metal counterstain that we use to reveal tissue morphology. And to the right, you can see these large bubbles, and these are actually fat cells. Or to the lower left, you see this round structure, which we think was once a duct that now is filled with tumor cells. So now we can lay the marker expression as false color on top. And what you now can see on this slide are, for example, small red dots. And these are actually cells that undergo mitosis because they're high in phosphorylated histone H3, which is a mitotic marker. Now on this slide, you can see two different cytokeratins, cytokeratin 5 in blue, cytokeratin 818 in green, and pancytokeratin in red. And in healthy breast, cytokeratin 5 and cytokeratin 818 delineate different lineages of epithelial cells. But please note how in the lower left area of this image, these two markers are incredibly heterogeneously expressed in those tumor cells. And that really made it clear to me for the first time visually what tumor heterogeneity actually means. Also looking at other markers, here we also have an antibody that gives us an epigenetic readout. Also here we can see great cell phenotype heterogeneity. But another aspect that I want to highlight is here you can see in green CD45, which is the pan immune cell marker, and CD68, which can be a macrophage marker. And please note how there are a lot of immune cells in the center area and the lower right area of the image, but most macrophages are actually in the upper left area or quadrant of this image. And this also makes it clear that within such a tumor, there's not only a great cell phenotype heterogeneity, but also an enormous spatial heterogeneity in where different cell types are and how they then interact with each other. And this then obviously begs the question, how can we analyze these data? And over the last few years, we developed a toolbox that we call MyCAT, which stands for Multiplexed Imaging Cytometry Analysis Toolbox, or in short MyCAT. In MyCAT, we currently have 4 modules implemented. In the first module, we can process the images, we identify individual cells, the neighborhoods, but we can also do gating on the images. Then we can push those selected areas into our second module, where we have different algorithms to study which cell phenotypes we have present in the images. In the third module, we develop novel algorithms that on a global scale allow us to analyze cell-to-cell interactions and microenvironments. And in the fourth module, we can then relate the cell phenotypes and cell-to-cell interactions with clinical and patient data. And what I can also announce now is that we have a stable beta version. Should you be interested in testing this toolbox, we are happy to share MyCAT with you. And I also would like to stress it not only works with imaging mass cytometry data, but also with serial chromogenic or fluorescent imaging approaches. The last point is that MyCAT is really built in a user-friendly and intuitive manner. And when you, for example, select a given cell type, you can readily see where those cells are located in the tissue. You can see their neighborhoods, or if you select a neighborhood, you can immediately see which cell types are part of that, and again, where these are located on the tissue images. Now, what is the current status of imaging mass cytometry? I told you that routinely we need about an hour to image a square millimeter. However, we can push the approach a bit, and here in my group at least, we can also measure square millimeter in about 30 minutes. The resolution, we routinely use a micrometer spot size, as this is for us the sweet spot for throughput, sensitivity, and resolution of the cellular compartments. However, we can readily generate images at 500 nanometers or even at higher resolution if we feel that it's needed for a biological question. We can measure 52 markers simultaneously, and here it is important to stress that the technology itself already supports now the ability to measure 135 markers simultaneously. There's virtually no overlap among the measurement channels. I told you about the large dynamic range. We can use frozen tissues, formalin-fixed paraffin-embedded tissue, adherent cells. So we also use it for bone samples. So all the tissue types that are typically used in life science research, we can analyze by imaging mass cytometry. Sensitivity is probably in the range of 100 copies per pixel. I told you that we have about 150 antibodies analyzed. And the whole approach is plug and play with the mass cytometer. And also soon a commercial system will be available that can be purchased by anyone to do imaging mass cytometry at their own facilities or labs or company. On the last few slides, I want to give you a few examples of what we can do with imaging mass cytometry. So in one study, we are analyzing a cohort of breast cancer samples. For every patient, we have 2 areas of the tumor core, of the invasive front, of the available, of the metastatic sites if available, and the matched normal tissue. And of course, we have a lot of clinical information available in addition. So then typically, we first look at the cell phenotype. And here I would like to stress, on this slide, you do not see a tissue section, but what you see is a projection of the single cell phenotype from the high dimensional in 2-dimensional space using an algorithm called t-SNE. And to explain this to you, every tiny dot is an individual cell, and if 2 cells are very close to each other, it simply means that the marker expression was very similar. And the marker expression shown here for estrogen receptor, HER2, cytokeratin A18, and eckaterin can be then shown in a color scale, where here blue is lowest and red highest expression of a given marker. Now, what I found truly fascinating is there were 3 clusters, 3 cell phenotype clusters driven by 3 patients that were all classified as luminal HER2-positive patients. But if you only look at estrogen receptor, you can already appreciate that it's lower expressed than in patient 1. Then if you looked at HER2, patient 2 has lower levels in patient 1 and 3. The same is true for the cytokeratin. And ekaterin is much higher expressed in patient 3 compared to the other 2 patients. And when you look at all the other 40 something markers that we measured here, you would be surprised to see how different those 3 patients are from each other. And that is something that we now see over and over again. While the classification using just 2 markers and doing a kind of yes and no classification is already very useful in the clinic, and our technology nicely reproduces that. We did this many times. If you start to look quantitatively at those markers and many others on top, you actually see that these tissues and therefore tumors can be actually quite distinct from each other. So we can then also use clustering algorithms to identify cell phenotype clusters. Here we used the phenograph, and we identified 29 cell phenotypic clusters in the cohort of these breast cancer samples. And in the next step, we want to know whether we can identify patterns of cell-to-cell interactions in those tissues, or to rephrase it, whether there are certain microenvironments, whether there's certain structure in the tissue that we can identify. And for this, we developed several algorithms. They're also implemented in MyCAD that allow us to perform a systematic neighborhood analysis over all the cell types that we can define using algorithms or also manually. And what we do is that for every single cell, we determine its nearest neighbor, so you can also define a radius. Then we do some statistics of which cells we actually find, and then we develop algorithms that give you as an output that certain neighborhoods are enriched in the tissue, or that certain cell-to-cell interactions are actually avoided in a tissue, which is as important to describe the structure of a tissue. And we did this here as an example for 52 patient samples, where red shows the interaction and the blue the avoidance of 2 cells being next to other. On the x-axis, you see all the combinations of all cell phenotype clusters versus all phenotype clusters. And on the y-axis, you see the individual samples. And in white, you see tumors that were grade 1, and in black, tumors that are grade 3. And you can readily see that these actually separate according to the clinical status. And when we look closer, we can see that this is driven by the actual cell-to-cell interaction structure that we have identified in those images. Such a heat map is very informative. It gives you a grand view on the data. But we typically then transform this using also a set of algorithms that we implemented in the form of a network, because that's easier to interpret. And now what one can readily see is that the grade 1 tumors, they show a lot of structure, as there's a lot of cell-to-cell interactions and a lot of cell-to-cell avoidance, while the grade 3 tumor has much less of this type of structure. Then when you look closer, we see here this node number 3. That's actually a tumor cell phenotype, and you see that it has a very strong interaction with itself. And that makes a lot of sense, because in the grade 3 tumors, the tumor tissue is still very well structured. So these cells interact readily with each other. And also the interactions that we find in this network are often with other tumor cell types. Now in the grade 3 tumor, you can appreciate that the cell phenotype number 3 does not show any more interaction or avoidance with other cell types. Now what is also interesting in the grade number 3 tumor is the cell cluster number 7, which is a tumor-associated macrophage cluster. In grade 1, we didn't find any significant interactions. However, then here in the grade 3 tumor, we see that there's structure in where those cells are actually located within a tissue. And for example, cluster 14 that you see then to the lower right to cluster 7, these are cells that are in highly hypoxic areas, and we do not find the macrophages being close to that. Now, I could explain you a lot more about the biology that we identified here, but the point that I want to make is if the algorithms that we have now developed, together with imaging mass cytometry, we can perform a comprehensive neighborhood analysis that reveals the structure and structural differences down to the cell type and the cell-to-cell interactions. And then we're only a single step away from using this data to stratify patients according to clinical data. And also here we implemented different approaches. The example that I show you here is based on the citrus algorithm that was developed by Rob Bruckner. And then we compare here the grade 3 with the grade 1 tumor images. We actually find that unique to grade 1 is again a cluster of these tumor cells, again with specific spatial properties. And the cluster that is unique for grade 3 tumors is actually high in CD60 and augmentin. So these are again the macrophages of which you have seen before, also that have a unique microenvironment. And what we strongly now propose is that we should really use the cell types and cell-to-cell interactions as biomarkers to classify patients for the subsequent treatment, etc. Now, where do we go from here? I told you initially that a tumor is a highly complex entity. And in a moment, we only analyze slices, meaning we generate images. However, to understand a given cell phenotype, I'm convinced that we also need three-dimensional information, because the red cell that I've highlighted here with arrows, we might only understand its cell state if we know about the cells above or below this cell. And therefore, what we have now developed in my group is what we call mass tomography. And so we use the imaging mass cytometry approach to generate three-dimensional volumes of tumors. And I hope that you can actually in the moment see the animation of this tumor. At the same time, okay. Now, in addition, we then also have developed a computational toolbox, which we call MyCat 3D, that allows us to generate those 3D volumes from imaging mass cytometry measurements, and also then to do data analysis in those three-dimensional volumes. And just here quickly another video where we generated a larger volume, again, of a breast cancer tumor. Okay, and then you could actually see that we have the resolution in the voxel space to see individual nuclei and cells so that we can really do 3D segmentation of the cells and therefore then truly understand the environment of every cell in 3 dimensions. So now coming back to antibodies. Validating antibodies is an enormous effort. It takes a lot of time, sometimes a lot of nerves, and obviously a lot of financial resources to validate dozens and dozens and dozens of antibodies. And we eventually thought that it is a waste if all of this validation data isn't only kept within a given laboratory. And therefore what we have developed is a database that is called AirLab. If you go to the AirLab website, you can make your own account. And AirLab has two important sites. There's the private site that is for the laboratory only. You can organize all your antibodies. You can do antibody purchases. You can tell which label your antibodies have. You can make panels of antibodies for mass cytometry, for flow cytometry. So you can really organize your whole antibody library with AirLab. But for me, the most important point of AirLab is that to every antibody, you can attach validation data. So you can say, here's the titration against positive and negative cells, or here's the experiment where we overexpress the marker. And all of this data you can attach to an antibody clone or a batch of antibodies that you have labeled. And upon publication, we hope that people will make this information available to the community. And we made this very easy because there's basically a button where you can say, make public, and all of the validation information about a given antibody is shared with the scientific community. And so in the medium term, we truly hope that AirLab will become something akin to an antibody Wikipedia. And we truly hope that it will save a lot of time spent on antibody validation in other labs. So also just quickly about antibody validation, we found that the metal labeling hardly ever affects antibody specificity. If any, it destroys the antibody altogether. We always follow the rules of validation that were outlined by Dr. Wetzel in the last presentation. It's very tedious to do all of those experiments, but you all know the saying, garbage in, garbage out, so better make sure there's no garbage going in. We readily use immunofluorescence microscopy and AHT to pretest and validate antibodies with high throughput. And once we see an antibody is very promising, we label it and push it to the revalidation with imaging mass cytometry. Then there are also many rules to design a panel of 40 antibodies to get truly high quality data. And obviously, one always needs to have many controls. The right controls to validate antibodies, but always having many controls that validate the performance of the antibodies when you actually do an experiment. And with this, I would like to come to the acknowledgement. Here's a picture of my team. And people say they're only standing on the shoulders of a giant. I'm standing on the shoulders of my whole lab. They really do a fantastic job. And about half of them now are work in the direction of imaging mass cytometry. The other half is working in the direction of suspension mass cytometry. And simply thanks to all of them for their wonderful job. And then I also want to acknowledge my collaborators and funding agencies, and here especially the lab of Detlef Günther, who initially to start imaging mass cytometry was a key person. And then I also want to greatly acknowledge the university hospitals of Zurich and Basel for their support in terms of the samples. And here's specifically Susanna Varga, and especially Walter Weber, who is at the University Hospital of Basel, and an incredible supporter of biomedical research. And I also want to acknowledge the Swiss National Science Foundation, the European Research Council, and the NIH as being the main funding agencies of my group. And finally, also the PATH Foundation, which is a tissue biobank that was founded by patients that actually want that their samples are used for research. And that's an amazing foundation, as they're incredibly supportive to make available sample collections and clinical data. And with this, I'm done, and we are happy to receive your questions.
Thank you for that informative presentation, Professor Bodenmiller. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can, and we will answer any remaining questions by email, including questions that are submitted after the live event. Okay, our first question is, given the instruments are not easily available, can you comment on the difference in throughput and sensitivity between your method and the multi-omics?
So I assume the reference to the multi-omics approach is the one from GE Healthcare. And so the multi-omics approach is based on serial fluorescence microscopy. And There are several advantages of serial fluorescent microscopy images of tissues, and there are several disadvantages. Let me start with the advantages. The advantage certainly is the throughput, because in this approach you label 1, 2, 3, there's now one publication with 5 antibodies at the same time, and then you can take a slide scanner and image really large areas of the tissues. That's an enormous plus. The caveat of the approach is that it's a cyclic method, meaning that you have to add the antibodies to the tissue, you have to image, you have to remove the tissues, the antibodies. Then again, you have to stain, image, again remove the antibodies. And it has been seen that due to the cyclic nature, it's very time consuming, and many epitopes lose the antigenicity, the morphology of the tissue can change. So these are the main challenges of the serial fluorescent microscopy technique. However, and also the dynamic range of fluorescent approaches and the autofluorescence of a tissue. But for me personally, the way I see it, having a technique like serial fluorescent microscopy, I see in the moment as a great tool to get a good overview of a tissue, already very informative, and then zoom into the most informative parts of the tissues with imaging mass cytometry to get the highly quantitative data with the simultaneously stained antibodies on the tissue.
Okay, and this one is for you, Bern. How is size measured in the ion cloud?
So when we analyze tissues, we do not measure any size. When you analyze individual cells, then the length of the ion signal that you can detect is roughly correlated to the size of a given cell. And we now also have reagents established in my group that actually give a very accurate readout of the volume of a cell. But we haven't published that yet, but that should soon be out, I hope. Keep fingers crossed.
Okay, and this is for you too. It is great to stain sections with 52 antibodies. Is there any saturation limit for number of antibodies cells can take or antibody-antibody interaction?
So that's an excellent question, and the question I frequently hear. So I cannot give a quantitative answer to that. What I can say is that during the validation process, we look at individual antibodies, at few antibodies, and at all the antibodies at the same time. And if there were steric hindrances or influences, then we would, for example, see that the signals of all the antibodies would go down, of many of them. And that's not something we have observed yet. And also in the tissue section, one has to think that it's, I exaggerate, but it's a two-dimensional entity. So there's a lot of space on top and to the bottom in where the antibodies can be. But then also after antigen retrieval, I think a tissue often, if you really look into it with an electron microscope, looks rather like a Swiss cheese than a closed tissue. So I think there's a lot of space for antibodies. Having said that, we have seen steric hindrances for antibodies that target the same protein. For example, phosphorylation site and the total protein. And there we actually can see such effects, but not over the whole panel in general.
For the laser-based sampling, are you going to get one single cell or cluster of cells? And if the samples are composed of multiple cells, how to accurately put every single cell to its right location during image reconstruction? So the resolution of the laser is such that we have subcellular resolution. So a typical tumor cell, I would say, is probably about 20 micrometers in diameter, or even sometimes larger. And our spot size of the laser is about a micrometer. That means that we measure for every individual cell about 400 pixels at a minimum. And the reconstruction of the images is done in a way that we know the coordinates of every individual laser shot. That is
recorded. It's a bit oversimplified, but that's in the end what it's done. And afterwards, we can simply use these coordinates to generate a grid, which reflects every single laser shot that was performed on the tissue. And then we can fill into each spot of the grid the information about mark expression. And that's when we have the image of the tissue. So that step is actually extremely simple.
Okay, are these stained FFPE or FF tissue slides?
So, as I mentioned in the presentation, the technology itself does not care a lot about the sample type you offer it for imaging. And what I showed was formalin-fixed paraffin embedded tissues, where we then had to do antigen retrieval, etc., etc. But we also image frozen tissue sections or adherent cell lines, or even cells that you stain in suspension, you attach to a surface. So all of this we can analyze. It's only about binding the antibodies to your sample of interest, and that is something independent of our technology, really.
Great. Okay, bear with me 1 second. I'm just grabbing another question for you.
Mm-hmm.
It's just a practical question. When IMC is used for FFPE, do you need pretreatment steps like antigen retrieval?
Yes, that is correct. We do higher retrieval, so heat-induced, and we do it at basic pH. And this already touches the topic of how to generate an antibody panel, because as Dr. Wetzel already pointed out, antibodies have certain optima, also in terms of the antigen retrieval. And in imaging mass cytometry, you have to find your roughly 50 antibodies that all perform pretty well at a single antigen retrieval condition.
Well, that will have to be our last question for today, but I'd like to remind everyone that we will answer all the remaining questions by email, including those that are submitted after this live webinar. I would like to once again thank Drs. Bodenmiller and Wetzel for their presentations. Do either of you have any final comments?
No, thank you. I would just like to say thanks to everyone for listening in today.
And also, I would like to thank everyone for joining this webinar, and thanks for your really good questions.
Well, thank you both, and I would also like to thank our sponsor, Cell Signaling Technology, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you, and we will see you here next time at labroots.com. Goodbye.